This is a lecture covering a couple of advanced topics. I'll have to go through them pretty quickly. I realize we're late in the course and everyone is tired. I'm more tired than you are. It turns out there's more jet lag involved in staying home and coming in by video link than there is by traveling. Anyway, the two topics are morphometric methods, which is a joint project with Fred Bookstein, and threshold models. You've heard some references to them, and there will be an exercise on them after the lecture. So we start out with morphometrics, and this is, I always wondered how you could use data from morphometrics as quantitative character data, because it is coordinates, but there are some complications that I'll bring up in a moment. The work is joint work with the guy who's really the biggest person in morphometrics, Fred Bookstein, who was for many years at the University of Michigan. And now he is for some years now in Seattle in partial retirement. And also he's been going back and forth to a job he had for a while in Vienna. Anyway, there are photographs. We figure we must have had a common ancestor, so we determined what the phenotype of the common ancestor was, which is JFL Bokenstein. I can tell people afterwards, I don't want to take up time now, how you do that, how you merge two photographs of I now know. Okay, well, the morphometric data, and here we'll mostly talk about two-dimensional morphometric data, but the similar issues apply to 3 dimensions. And I'm going to be talking about landmark data, where you're not taking a continuous curve, you're taking coordinates at known points, at sort of corresponding points around a shape. And you've got those as XY points. And you might think, well, okay, we collected 20 landmarks, that's 40 numbers. Surely we can just say those are quantitative characters and feed them in to the machinery. But there's a hitch. If you had, say, a nearly two-dimensional fish and you were digitizing it, there is the question of where you placed it on the digitizing surface. And that's not a property of the fish, it's a property of your decision as to where horizontally and vertically the fish is to be put down. So that affects all those coordinates. In addition, there's the effect of the rotation of the fish. You also made decisions about rotation, and these are not properties of the fish, they're properties of you. So the question is first of all, how do we get rid of the arbitrary contribution to all this by location and rotation? And in effect, it's asking about superposing specimens, but in a moment you'll see that superposition is, in a sense, not the central issue. Here, for example, this is a point where, a way to make the point. Here's the outline of a fish, and we have 13 landmarks around the fish, the places where the lines bend. And let's imagine 2 lineages. We start with the same exact fish, and it evolves up one lineage into this one, and it evolves up the other lineage into that one. And in this fish, the back 4 landmarks are pushed backwards 1 centimeter. And on this one, the front, actually it's 9 because I left out a couple of arrows, 9 landmarks are pushed forward by 1 centimeter. Now the question comes up, are those different forms? Anybody have, anybody have an opinion on that? Are those different forms? I can't actually hear anything anyone is saying. Maybe they're not saying anything. Well,
I think they're different.
Okay. I wanted somebody to be wrong, so you have helped a great deal by being wrong. No, these are in fact exactly the same form. Now, if you collected their coordinates, they would come out different. But all that's happened is that the distance between this part of the fish and that part of the fish has gotten 1 centimeter greater in both cases. But it's just that we plop the fish down differently. I know that these two forms are the same because when I drew the figure, I simply picked up this form and moved it over here and dropped it down. And that raises the point that what is a property of the fish is the difference between, say, this landmark and that one, but not exactly where either of them is given that amount. It doesn't matter where either of them is on the page. These two are, in fact, identical fish. They're different superpositions because you can see the gray here, which is the previous fish, and they're superposed differently, but they're in fact exactly the same form. So answer is no. And one way we get rid of that problem is by considering the differences among the horizontal coordinates and the differences among the vertical coordinates, but leaving out the grand mean of the specimen, which is just arbitrary. It's where you put the fish on the surface. And it amounts to taking contrasts, in this case, not phylogenetic contrasts, but between landmarks. And one way to get rid of it, that's the one we'll be using, is to center each specimen so the average of its points is at 0, 0. Its centroid of those points is at 0, 0. That has the advantage that you cannot alter the points by shifting the whole fish because you're not allowed to do that. So it drops 2 degrees of freedom, and now instead of 2 p coordinates where there are p landmarks, you have 2p minus 2, and the whole thing sort of lives in a small space. Now, you might think you could do the same thing with rotation, that there is some magical way of getting rid of rotation. That way we can, what I just did is what are called Helmert transformations technically. They get rid of the issue of location. Getting rid of rotation is much harder. But what we'll be doing is using different methods of superposing the fish, including one that is done with maximum likelihood machinery. One of the most common ways of superposing two sets of landmarks, two forms with landmarks, is what's called the Procrustes transform. And here I'm just doing it with triangles. And what you do is you take the corresponding landmarks and you connect them by lines, and then you take the sum of squares of those lines, and you rotate and translate the forms so as to minimize this sum of squares, which is called a Procrustes distance. And there is an algorithm. It's fairly easy for doing it. I won't go through the details except to say you take an average of the forms in their current locations and rotations. You then do a Procrustes superposition of each form to the average. Then you retake the average and do it again, and you keep doing this, and very quickly it converges on an overall Procrustes superposition of the fish, a least squares superposition. Notice also it doesn't use phylogeny in any particular way. This is all part of a procedure that's called the Morphometric Consensus. And it is very popular among people doing morphometrics. It's the central machinery of the field. We're actually going to be getting rid of it in this talk, but it was invented with some wonderful theorems by a famous probabilist, David Kendall, and using some machinery for principal components of distance matrices that were invented by this guy on the right, John Cower. And basically what they said to do, let's see is to superpose all the forms in what's called the generalized Procrustes analysis. And then you compute all the pairwise distances, the sums of squares, or you can square root them. So you compute a distance matrix between them. Then you use Gauer's machinery to get principal components of this distance matrix. It's called principal coordinates. And those coordinates are then the things that you analyze. And there's wonderful theorems by David Kendall showing that this is the right thing to do. But there's a problem with those theorems, the problem being they assume that the error in the forms is independent measurement error, independent in that each coordinate just has a circularly symmetrical measurement error. They're all equal and they're all independent. And that's an okay first cut model for measurement error, but it's not good for a model of how forms change along evolutionary trees. So we're going to do something different. And the problem that this morphometric consensus method has is that it's subject to what's called the Pinocchio effect. So here we have a picture of Pinocchio, and he's got landmarks, and he tells a fib and his nose lengthens. And if you do a Procrustes fit of those two forms, what you'll find is there's a little bit of error, a little bit of difference in all these forms in order to accommodate the nose. That the lengthening of the nose has meant that the Procrustes superposition will react in all the other coordinates because it doesn't want to accumulate a big square difference in the nose. What we like to do is have a way that did a superposition that's more like this, where you can see what's changing is in fact the nose and not everything else. And our method will sort of do that. There's also differences in the way size is treated. In the morphometric consensus, you take the size of a form is the sum of squares of the square root of the sum of squares of the distances of all the points from the centroid. We're going to have a maximum likelihood machinery for size. Now, in doing this, once you have your forms superposed so as to get rid of these location problems and rotation problems, we're going to be analyzing them with multivariate normal models. We're going to be saying that the points all move by Brownian motion, which is not perfect. Points could pass each other, the form could become very weird. But for small amounts of change, it's not too bad. For small amounts of change, you can take a process which really ought to be considered non-linearly, and you can approximate it by the slope of lines passing through the current location of points. And you can say, well, if a coordinate x is varying normally by a Brownian motion, then to first approximation, z here will vary normally in Brownian motion. Of course, the nonlinearity in the longer run becomes important. So we're sort of using a short-term approximation and saying, well, it'll be good enough from a sort of Taylor series point of view to consider small changes and consider that those are somehow linearly, that whatever's really changing is linearly affecting the points that we're measuring to first-degree approximation. And what we're going to do is to compute likelihoods. One of the methods we use is to compute likelihoods, and to do so with likelihoods on a phylogeny using the standard methodology that we've been talking about here. And we're going to use it in doing things like the rotations, and then also in inferring the sizes. I'm not going to go into how the whole size thing works because we don't have time. But basically, we're basically going, instead of doing a generalized Procrustes least squares fit, we're going to take the specimens, we'll get their centroids all the same, and then we'll try different rotations. And for each rotation, we'll go through the whole likelihood calculation on the tree and figure out the likelihood. And we'll come up with a maximum likelihood set of rotations. So when you do that, you basically have dispensed with the morphometric consensus. You're not computing a distance matrix. You're not going off to principal coordinates of that distance matrix. You're just superposing the form centroids, maximum likelihood analyzing their rotations, doing a similar thing to a sort of stretch factor that becomes the size, and ending up with a bunch of variables, shape variables and size variables, that you then just use this Brownian motion, multivariate Brownian motion model, where things are changing, but of course in a correlated fashion. And the individual points are not just subject to measurement error. Some might be changing much more than others. And that's taken into account in the model. Here's a simulation test to show you what results. What I do is I have here an imaginary fish with 10 landmarks, and the fish is changing, is being evolved up a tree with the following. The individual landmark locations are going to wander by Brownian motion that's circularly symmetrical. It's equal variance up and down and sideways and no correlation. And all those wanderings are going to be assumed independent. That's not quite the same as the morphometric consensus assumption of measurement error, because the changes are going to be cumulative up a tree. And in addition, we're going to have a component, which is the fins, getting this fin going up and down, and this fin lengthening at a 45-degree angle in a perfectly correlated way. So we have the individual points wiggling, and then we have these 2 points changing in a perfectly correlated way. So this is just a simulation we've done. And choosing some, choosing some parameters for that, we can evolve a bunch of forms. And we're also, I should say, let me go back for a second. I didn't write it down here, but we're also having the sizes of the specimens, a sort of overall expansion factor, evolving according to in this case, an uncorrelated log normal distribution that's going on it's a Brownian motion on a log scale that works out to a log normal Brownian on a scale going from 0 to infinity, because sizes can't be negative. When you do that, here's what we get. We're going to evolve 100 fishes up a tree. The tree was just randomly simulated. Here are 20 of them superposed, and you can see they differ in size, and various parts of them appear to be moving. So I'm comparing 20 of them because showing all 100 would be too dense. Well, when we do this process, we can also, we're inferring sizes when we just show the shapes, which have the size, standardizing them all to the same size. You get a somewhat better superposition. Looking at with your eyeball, you can see that something is going on here and something is going on there. It looks like something is going on here, but rather less at some other places. But let's see feeding the rotated and sized coordinates plus their size variable into a standard contrast machinery on the true tree. What happens? Well, what we get are, let's see, a first principal component. We get out principal components. And the first principal component we get out looks like this. And what these red arrows are showing you changes, and the heads on the arrows show you what kind of correlations we see. These are one principal component, so they're all perfectly correlated with each other. And you sort of see some signal. It's not really the one that we talked about. Here, if we standardize their shapes and just ask what's the, for this data set, which is one of 100 data sets that we generated, we do recover for the first principal components looking at shapes, the motion of the fins approximately correctly. The other points appear to be bouncing away in the opposite direction. And that's really because we're fixing the fish's centroid. So when the fins lengthen, the centroid shifts. If there's a way we can get, sorry. Yeah, here. There's a way we can make that a little more reasonable by not insisting that the centroid position be maintained. I think I will not describe this except to say it's a way of basically making the first principle component more sparse by mixing in a little location information. And when you do that, you really recover very nicely the main change of the fins that we were hoping to find. If you use morphometric consensus, things are not so good. And that's because you don't choose your rotations by maximum likelihood, you choose them by the generalized Procrustes. But in addition, you don't use the phylogeny. If you just take your specimens and put them together with, superpose them by the by the generalized Procrustes machinery, and then just compute the covariation without thinking about a tree, you get some of the same signal, but you get a lot of other stuff mixed in. So use the phylogeny is an important message here. Now, just to sort of finish off on the morphometric stuff, all of it is sort of cast in a very geometric framework, but it's good to remember that it would be nicer if we understood some of the genetics and development of these fish and knew, therefore, what the underlying variables really were. So we have a geometric morphometrics, that's what the field is called. We now have a phylogenetic geometric morphometrics. But we don't really yet have the genetic and developmental morphometrics that we would like to have. When we have that, then we'll be dealing with variables that are much more fundamental and will extract information more efficiently. And we can't do anything much about it right now, but I think it's important to keep that in mind as a long-term goal. And of course, the research program that we're thinking of is the same one we've been talking about in other aspects of the course. And one of them is to say, let's look at what we know about quantitative genetic variation and then look at evolutionary change among species and see if we can see the signal of, in addition to the genetic covariation, the signal of the selective covariation, and to get some idea of where, what selection pressures are, what correlations between changes are due to those selection pressures. So that's the quick answer to that's the quick coverage, running very fast through how you deal with morphometrics and what some of the issues are. I think before I go on, I should take some questions on this because we're going to shift to something very different in a minute. So, do we have questions about how you deal with morphometrics?
My question is the landmarks have to be present? Like for the 10 landmarks, they have to be present on every single object that you looked at? Is that correct? Like you couldn't have one that has 12 landmarks in one and 8 in another?
Well, you could have a situation where the fish has a landmark, but you didn't record it, or to say the specimen got damaged in that region. We don't yet have a simple way of dealing with that. In principle, it can be dealt with. We're doing the easy case first, but it's computationally possibly tedious, but I can think of ways to use a likelihood framework to cope with that and analyze all those specimens. Other questions?
So during the development, probably we will find some modularity between parts of the individual, and that's going
I'm not hearing you clearly. Sorry.
Okay, are you hearing better now?
I'm hearing your voice, but it's very it's sort of.
Is it better now?
It sounds like you're underwater. If you speak Speak slowly, put gaps between your words and into the mic like that, and then I can hear.
So during the development, there is modularity between parts of the individual connecting different parts of the body.
I cannot understand your question. Could someone repeat this question? I have difficulty, with accents. I'm sorry.
No problem.
I'm going to be the translator. So I think what he's saying is between the parts of the organism, they're not all independent, they're in modules.
Exactly. So I would like to know, how do you deal with the correlation between traits of the organisms in these specific methods? Because now we have two levels of correlation. One that's between species and one that's between the traits inside of one species.
So he wants to know how you deal with the correlations between the traits which are maybe manifested in these modular clusters in the correlation matrix.
Yeah, I think that the method as it's posed right now is just like all the other phylogenetic comparative methods. So once you've taken care of these rotation and size issues, and those methods all infer the covariation across the phylogeny. And the phylogeny informs the method how much difference to expect in the same landmark in different species. But then the use of the phylogenetic comparative method gives you covariances between all your characters. So if we have, for example, those two fins changing in a completely correlated way, then the method picks up, it estimates covariance matrices using the tree, using the phylogenetic structure, and by taking principal components from those covariances, which is a straightforward thing to do, you hope that the first principal component will be one of the major modes of change, including the correlations among the characters. And in this case, it worked. So it is built into the method that we have these two sources of covariation, the tree and the covariances of the Brownian motion. And those covariances of the different coordinates in the Brownian motion are what you're talking about.
And the covariances in the Brownian motion itself, those are a reflection of the covariances among the traits.
They're reflections of the covariances of change in the traits. And those in turn are reflecting the genetic covariation and the selective covariation, the net outcome of both of those. And the models, what I just want to say is that the multivariate geometric morphometrics is a very sophisticated field, but up till now, in the hands of most of its practitioners, phylogenies have been left out. And this is trying to not assume just measurement error from one specimen to another, but accumulated differences due to processes modeled by Brownian motion. I should say we're not the first people to bring phylogenies in, but it's only been in the last 10 years or so, and in a modest number of papers that morphometricians have started to say, oh, maybe we should think about phylogeny. But we're also getting rid of this extraneous morphometric consensus machinery, which is a more controversial issue. Other questions?
So this is Steve again. So is there, in listening to you talk about this, doesn't seem to me that there's a big impediment to making a quantitative genetic version of the model you're using here. Is there?
No, I mean, not at all. It's just like any other set of phenotypes, the measurements you collect. The different thing about geometric morphometrics is they have some geometry, and so issues like location and rotation come up and have to be dealt with. Beyond that, they're just a bunch of numbers. They're phenotypes. They're affected by multiple loci. It's all the same as in other phylogenetic comparative method tests.
So we'd need a G-matrix version for landmarks and a beta vector for landmarks, and we're there.
Yeah, I mean, you could, for example, use these methods as part of a breeding design. You could have half-SIB families and use multivariate morphometrics to make sure the rotation and locations and, and the size is somehow, made visible. And then you just go calculate G matrices.
Or just assume we have them and put them into the...
Yeah, because when you have a fish and it grows a protuberance on its forehead, well, some genes are doing that. That's quantitative genetics. Okay, yeah. Other question? I can hear you, I think.
Okay, I turn it on. Well, thinking about constructing covariance matrices with Procustis distances, don't you have the problem of spreading the variance of the most variable landmarks to all the others, and then you will confound covariation? Don't you have that problem?
In the standard morphometric consensus, that does happen. We're not using that, and we're not using the Procustis distances. We're doing our rotations by maximum likelihood, and our locations are taken care of by the mean, standardizing the means. It is true that the form that you're looking at has it has 3 fewer degrees of freedom, two location degrees of freedom and a rotation degree of freedom. So in a sense, if you have 10 landmarks, you don't have 20 points, you've really got 17 points. And to some extent, that does spread the covariation around. It means that there are aspects of the covariation that you just can't get because of the way you collected the data. You cannot tell whether a fish got longer because the front end went forward or the back end went backward, because the data don't have that in them. So we're not using morphometric consensus, we're not using Procustis, but we are doing a superposition by maximum likelihood, and there is some loss. There's intrinsically some loss of information. It's not just that we failed to collect it, it's that it isn't there. Perhaps I should, unless somebody else has a question, I should move on to the threshold model stuff so we don't run out of time. Let me see, excuse me here. What? Okay. Sorry. All right. So the threshold models, a completely separate issue. We have traits often that are discrete traits. We might have, for example, a plate on the surface of an insect, and it has an extra protrusion on it. And if we treat that as a discrete character and say it's either there or it isn't, we have discrete characters. And those are legitimate parts of morphology. And the question comes up, how do you model those either in some phylogenetic way, but also hopefully in some quantitative genetic way? And right now, people analyzing those on phylogenies are mostly using parsimony methods. And they're using them not necessarily for ideological reasons, but because they don't have any option. And in some cases, these things can be measured quantitatively, and you can turn a discrete character into a continuous character. But there will be some cases where you can't. And the question is, do we have any actual statistical models for the evolution of these discrete phenotypes? Well, there is one that's actually getting to be fairly popular. And that's one invented by Mark Pagel in 1994 and picked up in a major way by Paul Lewis in 2001. And they simply said, let's treat the alternative states of this discrete character as if they were different nucleotides in the sequence. We just have discrete states, and we have a rate matrix for transition back and forth between these states, and we assume a simple Markov model. In Paul Lewis's version, where there can be up to k states, it's called the MK model. And this is a fine model, and lots of interesting things are being done with it. But I want to propose an alternative that I think has some advantages, and that's the threshold model. And it was invented by this guy, Sewell Wright, who everybody, as I hope, heard of, one of the two greatest of the three great founders of theoretical population genetics and a major pioneer of mammalian genetics. He worked on guinea pigs. This is his photograph in 1954-55 at the time of his induction to the National Academy of Sciences. And he's posing with his organism, a guinea pig, and some of his diffusion equation formulas written on the board. And there's been a persistent rumor that right after the photograph was taken, that he turned around and absent-mindedly started to erase the board with a guinea pig. He was an absent-minded professor type, and he always furiously denied that this had happened. People who had taken courses from him said that they had seen that happen on other occasions. And there's a whole elaborate scenario for how he ended up with the guinea pig in his hand at the right minute, and actually started to erase the board with the guinea pig. And anyway, in 1934, Sewell Wright was working on crossing lines of guinea pigs, and he had a character that he considered as a discrete character, and it's whether the guinea pig lines had 3 toes on the hind foot or 4 toes. He actually had 3 states of the character: 3 toes, poor fourth toe, or good 4th toe. I'm just going to talk as if it's 3 and 4. And when he was trying to fit the results of crosses of different lines and make predictions what their F1s would look like, he proposed the following model. He said, let us suppose that there's an underlying character, which in this literature has come to be called the liability. It's one we can't see. It's a hidden state. And there is a developmental threshold. And if your stuff on your liability stuff exceeds that threshold, your developmental process will make a 4th toe. And if it doesn't exceed it, it'll make only 3 toes. And he fit this model to crosses of guinea pigs, and it did a pretty good job. Well, what I've suggested is to take the threshold model of 1934 and use it and assume a tree and assume that the liability scale is undergoing Brownian motion. And it will be, you'll have multiple characters that will be correlated Brownian motion. So we're going to assume that along a lineage, you want this distribution of individuals. Here's a normal distribution of liabilities of individuals in a population. And we're going to assume the mean of that distribution wanders. And here's a particular simulation. And you're getting, the mean starts out at the root. I happen to put it at 0 at the root, and it wanders up and wanders down a little and wanders up again. And as the mean of the distribution changes, the fraction of individuals in the population that are state 1 changes. And that's shown by the shading on these lineages. Now, what's good about that model? It's not nearly as tractable as the Pagel and Lewis model. But what's good about it is it has some biology built in that the Pagel-Lewis model does not. For one thing, it has within species variation built in. There is assumed to be a distribution of the liabilities. We're not changing it, we're just shifting it around. But at any moment, you make a prediction not just of the state of the whole species, but of the proportions of 1 and 0 that are segregating, in a sense, in those species. So it has within species variation built in. It also has another property, which is that the changes behave differently over time than the Pagel-Lewis model. In the Pagel-Lewis model, if you change from 0 to 1, you, let's see, for you, no, 0 to 1. At any moment, there's a constant chance of going back to 0. And if you wait 1,000,000 years, there's still that constant chance per unit time of going back to 0. It doesn't change. In this model, the underlying trait is wandering, and as it gets far from the threshold, you get out into a region where it's not likely in the next little period that the distribution will cross back over the threshold. So it has the biological property that when you're near the threshold, changes of the discrete trait are common, but as you get further away from it, they become rarer. The species becomes more stuck in having just that one state, although there's always a possibility it will come back in the future. So to me, those have those are good biological properties to have. Now, how do we actually do anything with this? Okay, this is just the same thing I just said. The other thing I should mention is, whereas the Pagel-Lewis model, if you have multiple characters, if you have 10 different characters, and you try to put covariation of the characters in, it's like a 10-way chi-square table. You have lots and lots and lots of interaction parameters that you're going to have to bring in. Here, the covariation of the traits is more constrained. It's the covariance matrix of the quantitative characters, the hidden liability character, that are important. And so you have a single covariance matrix. That's all the parameters that you need for, in this case, covariances of evolutionary change for this model. So it is much nicer, it's much more nicely parameterized with respect to things like covariances. What I've done is, this is all in a paper in 2012 in American Naturalist, is to have a Markov chain Monte Carlo machinery. And basically what we do is we take the discrete states, the discrete traits, and the tree, again, we're given a tree, we assign liability values to the tips, somewhat arbitrarily at first. And we reconstruct liability values in the interior of the tree. And we go around this tree again and again and again, reconsidering those liability values. And we use what's called a Gibbs sampler in the interior nodes. Oh, I'm sorry. A Gibbs sampler in the interior nodes. And up at the tips, we use a Metropolis Hastings sampler for those who are into MCMC. But so with this model of Brownian motion of liability of means of the species on a liability scale, and then individuals chosen from, as a sample from it, we get this and we can do Markov chain Monte Carlo. And so you can reconsider, here I had 1.69 as an interior node. We do a Gibbs sampler step, it changes to 1.48. Here is a graphical depiction for a single character of the updates of the liabilities. Here is our liability scale. Here at 0 is the threshold. Since the scale is arbitrary, you might as well put the threshold at 0. Here are two species that have zeros. Here are the rest of the species that have ones. They're connected by a tree. And we go around giving liabilities to the interior nodes, and the liabilities place them on one side or another, their means on one side or another of this threshold. But then we go around and we reconsider that. So let's reconsider the liability at the tip of this branch. It moves. You see it was there. And let me go back and do it again. And it moved. And we keep doing that. We keep reconsidering the tip liabilities and the interior nodes. And they wander, but always in a way such as to keep the observed phenotypes the ones that we actually observe. So we can do this in a multivariate space. We have to transform the space to do it, and I'm not going to go into that. But it's all done internally in a program called ThreshML, which is designed for a future version of FileUp, but I've released separately so that people can use it. And going through and doing this Markov chain Monte Carlo, you can also look on the tree at any moment and empirically infer the covariances of the characters. And as you wander in this liability space, have the nodes update their liability values, you accumulate an overall estimate of the covariances of these traits. Here is a tree. Here's some simulated data simulating the threshold process on a true tree. We're going to do something similar with a 100 species tree. In a numerical example, we're going to have 100, let's see, no, I'm sorry, this was 50 species, I guess. This is 50 species. Okay, with three characters instead of 4. There's a true covariance matrix. We can simulate data on it, and we can then analyze the data with this Markov chain Monte Carlo machinery and ThresherML. If we do 2 different analyses, we'll find that the covariance matrices inferred in the 2 runs are in fact very similar. They really are seeing a signal there. Here is 50 trees, each with 2 simulated data sets. I think they're 50 species data sets. So these are like 50 evolutions of 3 characters. So they've evolved in 50 replicates. Of course, in real life, we only see 1 replicate. And then we run our machinery and we infer their covariances. And what we find is that the covariance between character 1 and character 2, it's very spread out. It's sometimes negative, mostly positive. It has, its true value ought to be 0.5357 for reasons that I won't go into. These are actually correlation coefficients. The true correlation coefficient should be 0.5357, and you can see it is varying around that. This one should have a true correlation of 0. It's very spread out, but again, roughly centered right. This one is actually, there's a missing minus sign there, minus 0.5145. Again, it's mostly showing negative values. So one lesson you can get out of this is that we are correctly inferring the underlying covariances on average, but there's a lot of noise involved. And that's really not terribly surprising. Because if you go to a statistician and you say, I want to estimate covariances, they'll say, fine, how many hundreds of data points do you have? And you'll say, well, I have 50 species. And they'll say, oh, that's painful. If you only have 50 points, you aren't going to do a very good job of estimating covariances of numerical values. And then you say, oh, no, wait a second. I didn't tell you. These aren't numerical values. They've been thresholded. At that point, the statistician may throw you out of their office. You are asking a lot to ask to infer these covariances very precisely. You just have, 49 degrees of freedom with 50 points, and you're going to have to accept that any inference you make from 1 replicate will be noisy. And you can see that, by the variation of the inferred values around the true values in this case. But there's some nice extensions you can do to this. One of them is somebody might say, look, I have data with some discrete characters, and I also have some continuous characters. How do I combine those in a common analysis? And for a while, I was mystified. And then suddenly it hit me that in this Markov chain Monte Carlo machinery, for the thresholded characters, we assign the liabilities and then we'll have them wander, keeping them on the proper side of the threshold. If we observe a quantitative character, that means we actually observe the liability. And the liability is the quantitative character. So maybe we could do the analysis with a mixture of discrete and continuous characters by simply saying for the continuous characters, we observe their liabilities and just fix them at those values. That's the only thing you have to do. And then you run the whole machinery and it works. It analyzes covariation between continuous characters, between each other, between them and the discrete characters. And it gets you out an overall covariance matrix. Now you may say, well, if we had only continuous characters, we don't need any of this machinery. We can use phylogenetic comparative methods, contrast, phylogenetic regression, whatever. And you can. And what I've verified by simulation, by running the MCMC is if you do just continuous characters, it gets you the correct values, you know, with some noise based on how long you run the MCMC, but it's getting the right thing. However, it's really useful for cases where you mix together continuous and discrete characters, and then you can do an overall analysis. And let's see, I've said all these things. Here is a case where we have 3 characters. We have character number 2 has been thresholded. It's a discrete character. Characters 1 and 3 are continuous characters. These are actually the same. They're the same 3 characters, except we thresholded not all of them, but just character number 2. And when you do that, once again, you get distributions. Once again, there's a fair amount of noise. These are the covariances, not the correlations. The red triangles show you where the truth is supposed to be. And, you know, roughly speaking, it's doing the right thing. So you can analyze a mixture of discrete and continuous characters by this machinery. I just want to finish off by saying that one extension to this that we have to think about in the future is, for all of this machinery, integrating it with the statistical genetic machinery of inferring quantitative trait loci. In the long run, statistical genomics is going to come in, and we're going to find ourselves looking for loci that have major effects on our quantitative characters. And there will be an era, and we're not there yet, in which QTLs are inferred not just in one species, but across a group with multiple experiments in multiple species. And you'll be able to ask some very interesting questions there. So I just wanted to say that look forward for this, to any of this machinery, and really everything in our course in the long run, also connecting to statistical genomics carried out on the characters. And so you should be able to infer not only QTLs causing variation in one species, but you'll be able to watch that change across species. And you might find some loci that are major sources of variation in chimpanzees, but a much smaller source of variation in gorillas and so on. And there's going to be a lot of interesting things you could do to that, including it does allow some ways of looking at 2 correlated characters changing in evolution, and not just saying, well, they're correlated, so selection could be on both of them, or on one, or on the other, and the response of both is due to the correlation, but we can't tell which one is really being selected. Looking at the QTLs, there will be ways of getting at the question of which one is under selection. That's a whole discussion of its own, and I'm not going to go into the details for now. Okay, I've got references here, and they unfortunately overlap the edge of the slide, but you'll be able to figure them out. Fred Bookstein, a book by him, Dryden and Marty, is the big statistical machinery book for the Morphometric Consensus method. Here's Gower's paper and another book on the subject. The methods we developed of doing superposition and rotation by maximum likelihood are very, very similar to methods that Doug Theobald at Brandeis University developed in this paper, Theobald and Wittke, and it's quite similar, but it has some important small differences, and on those points we're correct and he's not. Okay. For the threshold model machinery, here's Wright's paper, an extension by Douglas Falconer in the 60s applying threshold model to human diseases. It's been extensively, the threshold model has been extensively used by quantitative geneticists in animal breeding. Dan Gianola at the University of Wisconsin has written extensively on it. So we're not the first people to make modern statistical use of the threshold model. Here's Paul Lewis's paper, Mark Pagel's paper, and some papers in which I've either mentioned or developed the threshold model machinery.